Okay. All right. I think that we have 20 people now. I think we might have more people will join us today. Good afternoon, everyone. Welcome to our bioinformatics and genomics seminar today. And so Dr. Catherine Bernstein will co-host this meeting with me. Thank you, Kat. So as usual, we will talk about the RC wiki page. So this seminar is the second to last one, and we have one more in December 1st. So the speaker will be from University of British Columbia, who will introduce their lateral communication paper. And I think in this paper, they will introduce NIST database, which is very interesting bioinformatics resource. We might also potentially have one more speaker in the middle of December, and we need to confirm that. After we confirm that, we will update this page. So if you missed any previous seminars, you can also come here to watch the YouTube videos here. So with that, I will hand it over to Kat. Hey, Kat, can you?
Hi, thanks so much. I'm happy to introduce Jaroslav Budis from Comenius University in Bratislava, Slovakia. He's also been at Genoton since 2015 as a bioinformatics scientist, and a perusal of his publications shows that he is an expert in the field of non-invasive prenatal testing. And at Genoton, he leads a team responsible for bioinformatics research and development, as well as the implementation of acquired methodologies in clinical practice. Active multidisciplinary cooperation has led to the numerous publications, especially in the field of genomics, metagenomics, and bioinformatics. He's the co-author of 7 patents in the topics of non-invasive detection of chromosomal disorders in the fetus, interpretation of structural variants, and privacy of genomic data. In 2019, he became the principal researcher of the study of identification and genotyping of STR motifs with a focus on clinical diagnostics. He is also the National Research Coordinator for the European COVID-19 Research Data Platform Initiative. And I'm really excited I'm looking forward to his talk on the BioRXiv preprint, automated prediction of the clinical impact of structural copy number variations. I'll turn it over to you.
Okay, thank you, Catherine, for kind introduction. Welcome you all here. So I'll share my screen. No. Is it okay? You see the presentation, please?
Yes, we see it.
Okay, great. So I would like to tell you something about prediction of structural CNVs and how that can be automated to make our life easier, mostly for clinicians. Okay, so I will be talking about genetic testing that changed really things around here for last decades, and so we are trying to find something in the human genome, and new biomedical technologies allowed us to do that last decades. And from the clinical point of view, we are trying to discover some harmful changes. So something that changes according to some reference genome, and we want to find out what it is and how does it impact the life of an individual. So typical outline of the whole genetic testing is we take some sample and we digitalize it. Right now, the great technology are sequencer, either next generation sequencing on MiION, but in the end, they tell us something about the human genome. Next step, it's mostly bioinformaticians there that are trying to detect variation. So something that's in the sample that should not be here or should not be in a healthy individual. And the last step of the analysis is the interpret those variation. Either it's some single nucleotide polymorphisms, indels, short random repeats, or in this case, copy number variant. So So we are really focused on parental testing because parental testing is right now the most common genetic test in clinical settings, and we are trying to detect the changes of on whole segments of chromosomes, of whole chromosomes. So it's already established test worldwide. We are detecting mostly trisomies 13, 18, 21 because with those trisomies, child can survive and live a few years, but you'll get really severe defects, as we know, Down syndrome and so on. So it's a really important task to catch those pregnancies with those defects early. And one technology or method that changed revolutionized all this field was discovery of fetal fragments in the blood of mother. And due to this invention, it was now we are able to detect some structural aberration just from the blood of the mother. So we do not have to put mother at the risk to take amnio fluid by invasive methods. And these methods have already pretty high detection rate. So this kind of test is established worldwide. We have a huge experience in this area. We are testing for 8 years. We analyzed more than 10,000 samples, and we have a lot of publications there, either from laboratory side of biointerpretation side, how to improve those methods. We also filed some patents, so some of them are already approved. But basically we started with those three basic chromosomes, 13, 18, 21. Then we improved gradually those methods. We added sex chromosomal aneuploidies. They are more tricky because there are a lot more combinations. How could they combine those aneuploidies? We also include other chromosomes naturally. And as this laboratory processing improved and the price was cheaper, we could afford to improve even more those methods. And basically, if you are not looking at chromosome as well, but if you improve your bioinformatic methods and increase coverage, you can detect also smaller changes of DNA, some small segments of DNA. For example, at this example, you see the case of DiGeorge syndrome, that we see that there's a lower amount of genomic material at the area that it's associated with DiGeorge syndrome. So we can predict that. So we took some 5 recurrent syndromes, it's DiGeorge, Prader-Willi, and so on. We did some validation studies, we had some samples, and we developed a method for detection of them. So from technical point, we have really good detection methods to identify them. What was the problem that why focus just on those 5 syndromes when we can detect all of DNA, all of the genome right there? So the main problem about extending those tests even more is that the interpretation of the segments are really hard. If you think about that, we have 3 billion bases there, and each region can be affected. So there's a lot of combinations that can be affected, and the most of them, that we observe in a child or a mother are unique. They are new. We cannot compare them directly with some known genomes or some published cases. So this is really a problem because DiGeorge is, if it's affected in recurrent syndrome like DiGeorge, it's easy to tell the clinicians what do they have there, and they can consult mother. But if we have a new case, it's a problem. No, we cannot just tell mother that the child have something there, some CNV, and don't tell her any information how to handle that. Another problem, most of those CNVs are part of natural human variability, so they should not be reported at all. Why stress matter if it's something benign? And with it, there were some studies. GNOME did some global study. It was released this year. There's some really great structural map of different populations, and they found rare CNVs in almost 4% samples. So a lot of individuals that would have some findings reported. We did a similar study last year in the Slovak population. It was more or less the same. So what we need really, what we really crave for is some method that can tell that okay, this structural variation is not problem. Do not report and filter those that have that would be pathogenic and consult them with mother. So what we have right now, we have some expert decision system, some guidelines that were collected and by American College of Medical Genetics. They came out in last year, November, and really, those clinical evaluation of the sample should follow these guidelines to be supported by really experts in the area. And those guidelines are basically some scoring system. We've got 5 categories or 5 different genome characteristics you are looking for, and at each category you assign some option that's there, and some score is assigned. If you add these scores together, you've got a total score, and higher the total score, the more probable that the finding would be pathogenic. So here's an example of first category that's pretty simple. If it tells either it has some functional important elements or not, and so you choose one of the option. It assigns either 0 or minus 0.6 points, and then you can go to the next step. So what's the problem? The main problems with that guideline are several. First one is that besides category 1 and 3, the category 2 and 4 are really tough to solve. For example, here is category 2 where you have 12 options you can choose, and it's a lot of places you have to look to correctly answer what is present in that patient. You really need to look at really different characteristics in many different databases to get it correctly. Because if you go through these options, they're sometimes really tough to solve. And another problem is the variability of the score, because in some options, for example, here in 2C1, if you click those options, you can choose between 0.5 to 1, and if you choose 1, it's pathogenic. If you choose 0.5, it's uncertain significance. So it's really subjective to clinicians up there, and that's a problem because those guidelines should lead to reproducibility across different clinics, across different clinicians. So what were we thinking about was some automated prediction, because machine learning algorithms have proved to be really useful addition to solve those hard problems. And this is really hard problem to solve, to interpret those CNV findings. The machine learnings are really common in clinics. They are really common in oncology, in interpreting SNPs, indels, and so on. So we're thinking, okay, if we get all of the data that clinicians see and put it into some machine learning algorithm, would it be better? So we developed a method that was presented in an article on BioRXiv, and it's the idea is that we collect all attributes or characteristic of the genomic region, of the affected genomic region, that can tell us something about pathogenicity of the change. So, for example, we can take how many genes are there, and okay, we can collect the number. It's 36, and then we can look about known cases. How many genes were deleted in known deletions, and by some clever modeling, you can estimate some risk of this number. So in this case, we've got 8 attributes. Those red ones are really risky. You should look at them. And okay, those are medium. We do not know, and this one is a really low risk, so it's not a problem. It's really nice because it can focus you on that problems, on those characteristics that should be problem. But we can do better. We can aggregate all of those attributes. You know, we can get some map of that, and now we have better feeling. If you have more blue color there, then it should be more risky for pathogenicity. The smaller. So. And if you put those data to some machine learning algorithms, you can get prediction if it's benign or pathogenic. So machine learning algorithms are really great at that. If you get a lot of data and put it, pre-process them in some numeric form or something, it's that they can do really miracles for prediction. So the first step is how to select the attributes. So what was presented in publication are those types of attributes presented on the slide. The most obvious attribute is length of CNV, because the longer CNV, the more chance that it will affect some critical region. Should not be. With that highly correlated attribute is number of genes that are there, and we can look at total genes. We can look at morbid genes that are associated with some pathogenic impact. We can also look some scores like loss of function scores, kernel severancy, and so on. So we selected those 8 attributes that are presented here, and compare them. Compare the accuracy. So these gene attributes here, it is presented. The green line shows that we were accurate at 96% of the cases. So if 100 cases will come, 96 will get covered correctly, just by those 8 attributes there. And we compared it with one method that's available. It's classify CNV, and that classify CNV is heavily based on ACMG guidelines. It's actually trying to set out options on those ACMG categories automatically, and use the same scoring scheme. And if you compare them, they are they're more accurate. They're really accurate, but the number of predictions is not high. If you get there, it's only around 24% cases that they can tell some prediction. So how can we be better? The problem with if you look at if you look at our prediction that's presented with those box plots, you'll see that true negative scores are really below 0, as you would expect, and true positive cases are situated really around 1, as you would expect. And we have a lot of outliers here. And if you compare those box plots with false predictions, you see that there's a huge region where are mostly saturated by false predictions. So what can we do? We can really cut those regions and tell them that those are uncertain. We do not know what they are. It's similar that classify CNV, and then we have much better prediction power, around 98%. So natural next step is, okay, what if we give everything we got about the genome variation, not just genes, not just the length of CNVs, those obvious ones, but extend the set of attributes. So we worked on that since we published that on BioRXiv, and right now we accumulated more than 200 attributes that is telling something about affected genomic regions. And so extended about overlapping CNVs, if there are known cases about pathogenicity or about benign, they are included there. And there's also difference if you are using full overlaps, partial overlaps, and so on. We are using some established regions that are known to be problematic from haploinsufficient or triplo sensitive. If you're then another point of view are regulatory elements, because if you affect some promoter regions or some structural important regions like open chromatin, TIF binding, and so on, it could be problematic as well. And we also worked on those gene attributes. We added a lot of more attributes there. Where now we included a lot of prediction algorithm scores for loss of function of genes, the conservancy in population, but also conservancy with model organism or similar species. Some curative annotations there, known diseases, and so on. And the precision was really nice. Right now we are at a 90% accuracy, even if we do not have any uncertain predictions. So the next step that would come soon is if we allow some uncertainty for uncertain predictions, how would be the accuracy? But right now it's pretty killer, because the vast majority of the cases can be concluded accurately just by this machine learning algorithm. And it has huge advantages, because the process of interpretation of those CNVs is really cumbersome and really problematic. You have to look at many different genomic databases, a lot of sources, publications, and so on to make a really good decision. And you really need experts in this area to do it properly. So you got experts that needs a lot of time to do that, and that's a huge advantage of machine learning, because it's fully automated. You have just put position of CNV there, and all of it would be automatically annotated and interpreted according to those annotations. Another huge advantage about machine learning approach is that it will be continuously improved. As you see, the machine learning algorithms are as good as the data we put in them, and we have a boom of genomic data and known cases, and more and more genomic attributes. So if people would give more and more findings in public databases like CleanVar, Decipher, and so on, and we can use them and be better. If new genomic databases with new characteristics would appear, we can include them into machine learning and would be better. Each characteristic that can differentiate between pathogenic and benign CNVs can be included in this modeling. And then also machine learning methods are improving also. So there's no step back. With each iteration of this machine learning, with each new data set, we should be better at that interpretation. And the great thing is that this method is suitable for any detection method. We just need to see where is situated the finding. For example, we need to say that it's deletion of chromosome 4 on that coordinates, and it would be automatically annotated and interpreted. So how to really use it in clinical? Because it's really nice that we bioinformaticians can use it, but it's not the point. At Geneton company, we are trying to those findings. We are trying to put it into practice for the benefit of the patients. So we believe that both approaches are really helpful, and they should be both used commonly, because this expert system, these expert guidelines are really good for interpretation. They can really point at the problem that is in this data, and should be used. But machine learning is really useful to guide clinicians where the problem is, and give their own prediction that could be more accurate. So we believe that both of those approaches are complementary and should be used. So what we're working on is the project Genovisio, and it's some web application that allows clinicians to use the power of expert system and interpretation as well. And I would like to show you the current version of the Genovisio and where we're heading. And this project is right now is open, so if you have some findings and some CNVs, you can try it. We would be grateful for all feedback we can get. So you just need to go to Genovisio. There's link. There's some really simple registration there, and basically what we need to know about those regions are just coordinates and type. Maybe some identifier if you want to connect them with some of your case, but it's internal. And if you analyze after a while, you would get something like that. It's a clinical report, and it's heavily based on those ACMG guidelines. So you've got 5 categories there as ACMG. Each category has its score. You can write your own interpretation there, and at the end you've got some decision based on ACMG, in this case likely pathogenic. You can edit all of them because we are free sections we can automatically set. So we can automatically preset you that there are some protein coding or functional elements there because we annotate them. We provide you also some annotations that should explain you why we choose this option. So in this example, we got regulatory protein coding genes, other genes. We have some manual created regulatory elements there, external link that will lead you to publications supporting and genomic view of the region where you can see genes, you can see regulatory elements. The same, for example, established regions. If you are if you are evaluating section 2, you want to see if there are some upline sufficient triple sensitive regions. So in this example, there is such region. There is how is covered external link and also some supporting genomic view. And you can go through all of the sections. Some genes or some tables will give you supporting information about those genes and so on. So by filling one simple formula, you'll get full annotations of the genomic region. So what we cannot not do automatically is family history. It's based on your case, and detailed evaluation have to be assessed by yourself. But it's also problem with classify CND tool that this section is really hard problem to solve. But at the end, you've got your assignment of the points according to those guidelines, and you'll see that it's likely pathogenic, and you got in addition other score that says in this case the same thing, but it's up to you. It's two cooperative system, and you as clinicians should decide what's your so you can fill some text, finalize, and you will get a report that can be connected to the documentation. And the report is more or less the same as you've seen in the web application. Okay, so as I said, we would be really grateful for each feedback. You can get us for that. If you're interested, please try the applications. You get 10 analyses for free. And so I would really like to acknowledge all of the team that was working at that project, because we have people that are working hard on the improvement of this machine learning. There's a lot of space to improve even more. So we're working on a revised version of the manuscript that would have much better accuracy and would be released pretty soon. I would also like to thank all people that were working on the web application, because it's really tough to just get all those those genomic information at one place in some meaningful way, so we are not lost. And also would like to our colleagues from academy that guide us, and they're from the clinician side that are improving what is the need of the clinicians in there. So as I mentioned, this is a venture of company Geneton and also Comenius University in Bratislava and Select Academy of Sciences. So thank you for attention, and I would be more than happy to answer any questions. And if you would have some question later, you can write me on the email. It is right there.
Thank you. We have one question from Sophia. So Sophia, can you ask a question? I think that you basically just send the question to everyone.
Hi, can you hear me?
Yeah.
Hi, so thanks for the presentation. So one of my questions is that how much overlap do you need to have with a known copy number change to actually consider it the same and like a known CMV from Nomad or from ASAC or from some other database?
Do you mean this common population or just some published cases? Actually, to machine learning algorithm, we give partial overlaps, and we are giving them full overlaps, and we are experimenting about the threshold. What's the best? So right now we do not have it established. Current model is working with either any overlap or full overlap.
Okay.
And for that web view and those numbers that we are giving them, it depends on if it's a duplication or deletion. Because when you are interpreting deletion, you are interesting on known CNVs that are fully overlapped by your CNV.
Mm-hmm.
So it's
okay, makes sense.
And in deletion, it's otherwise. You need the benign CNV to overlap your CNV
Yes.
completely.
Yes.
So we are treating them differently according to the type of CNV.
Okay, thank you. And yeah, so my other question is that do you have an offline software available or not yet, or not at this point, or you're not planning to have something?
Yeah, we have source code there. Right now, this annotation step is okay. What we have completed is the interpretation, and we have offline code on GitLab, and it's accessible from this BioRXiv version. But it's just interpretation, so we would need to annotate by yourself. Mm. But we are working on that revised version where the annotation step should be also included.
Okay.
So at the end, it could be possible to offline, but not right now.
Okay, thank you. Thanks.
Sorry, just follow-up question. What's the input data for the software to basically automatically predict the CMVs? Is it...
We just need the coordinates, so chromosome start end of CNV and type. We can handle duplication deletion right now.
We have two more questions. One is from Christy. Christy?
Yeah, hi. I was just wondering if you had a minimum percentage of the fetal DNA for those NIPT tests. So a minimum fetal DNA that you would need for the copy number analysis versus looking at, just trisomies 21, 18, and 13.
Mm-hmm. It highly depends on, in trisomies, it's 5%. We enrich for fetal DNA by some laboratory size selection, but then we have established limit 5%. For structural, it's more tricky because it really depends on coverage and really depends on size of CNV you are trying to detect. So basically, if you have a huge duplication, a huge deletion, it's the amount of fetal DNA in the DNA mixture can be lower. But if we want to catch a decharge with 2 or 3 megabases, it's more tricky. We have some validation studies there. If you if you're interested, just type me and I can send them. But our detection limit was not sure about the exact number, but we can do from 3 megabases of dilution duplications and okay, maybe I can show you this heat map because this characters. Give me a second, please. Okay, so here's the detection method. So if we have 10% of fetal DNA and 3 megabases, we have a sensitivity of 93%, more or less.
Great, thank you.
It's comparable with 5% and 10 megabases.
All right. One more question from Celine Everett.
Yeah. Hi.
Oh.
So I was wondering. Oh, you said hello. Sorry, I didn't get that. I was wondering for the I had a couple questions. One was how long it takes for one case to be analyzed, and if this could be scaled to take large inputs from larger cohorts for research purposes.
How long does it take for one case? Yeah. It's a few seconds. The longest step is annotation. We have dedicated database just for the genomic data, and so we need a few seconds to annotate, and machine learning is pretty quick. So I believe when we did that on ClinVar data, it was around 20,000 or so cases, and it took 10 hours or so to get them all on one computer.
And does that mean you guys applied all the codes and it took that amount of time from the ACMG guidelines, or was that step not included in that?
Sorry, I didn't hear you well. So can you please repeat the question?
Yeah. When you say the annotation step isn't included, do you mean applying the ACMG codes isn't included?
Depends, because that interpretation of those categories, you mean that? Yeah. Right now it is included in this annotation, but it depends on it can change over time. So right now it is, but by the future it is possible that it would be missing there. It'd still be in the web application, but to get it automatically from the source code, it probably would not be there.
Oh, okay. Thank you.
So just want to double check. I don't think we have any more questions, but I have last question is that based on your presentation, so there's no way to use for example, NGS data, FASTQ files to predict CNVs. Is that correct?
You mean if you are using your FASTQ files to get interpretation? No, we have our special pipeline for our own laboratory processing, because we observe when you change some laboratory processing, you also change what do you believe is normal in the population. So the pipeline and configuration of those pipelines that can detect CNVs from FASTQ files are really dependent on laboratory processing. We can do it on our data. We can train our pipeline on some different data, but we wanted to do something that is universal. That each laboratory would have their own detection method that is not dependent on NGS or nanopore or anything. But at the end, they detect CNV and we interpret that for them. So that's the idea, just the step between detection and interpretation.
All right, thank you so much. I don't think we have any more questions. Okay.